Let's cover some nervous system histology. I have two slides to show you. This first picture, we're going to look at nervous tissue focusing on neurons. And then the next one, we'll look at a nerve. Remember, the definition of a nerve is that it's a bundle of axons. So when we look at it, you'll see that's very similar to how we organize a skeletal muscle. I've been teaching anatomy for about 24 years, trying to simplify this for you. There's also a study guide that can help you and organize these pictures to practice the identification. Remember when we looked at our different types of neurons, we said there's anaxonal, pseudo-unipolar or unipolar, bipolar, and then multipolar. Our focus here is looking at multipolar neurons that has that motor function. On the left side here, this is a expanded view. If I go into higher magnification, we're looking at just one multipolar neuron here. So as I look on the left, all of these big dark structures are the nerve cells or the neurons, the ones that are carrying out the neuronal impulse. All of these tiny little dark dots through the whole slide, these are all the supporting cells or neuroglia or glial cells. And so when I expand on one of these neurons and I pull out a multipolar neuron, that's one of these neurons here. So I can still see all these tiny little dark dots, and to me it's kind of look like space if you're trying to compare some of our slides. So all these little stars throughout, the tiny dots are neuroglia or my glial cells. Remember, there's 4 types in the central nervous system. There's 2 types in the peripheral nervous system. We had a previous video to talk about that. But as I focus on just then a neuron, remember the extensions of a neuron are dendrites and axons. And when we compare dendrites versus axons, dendrites tend to be shorter relative to axons, but axons tend to hold their shape, meaning they stay the same diameter throughout. Versus when I look at a dendrite, they tend to taper off. So some are more difficult to see than others, but this is a dendrite here because I can see it going wider and tapering off, versus an axon is a longer extension to it. When I look inside, remember all of this dark area is called the cell body or the soma, S-O-M-A, so cell body or soma. And then within it, I can see the nucleus, which is the rounder circle, the larger one. But then that tiny dark dot in the middle is the nucleolus. So as I look at neurons, we look at dendrites, the axon, cell body or soma, the nucleus, and then nucleolus in the center. And then all my tiny dark dots are looking at neuroglia. So this is nervous tissue focusing on neurons, specifically a multipolar. When I look at a nerve, now I'm taking this and I'm expanding all the different axons that we see in a nerve, because we said a nerve is a bundle of axons. And it's like a straw within a bigger straw within a bigger straw, just like we did skeletal muscle. As we learned endomysium, paramysium, and epimysium of skeletal muscle, now we're doing the same thing, that we have endoneurium that covers an axon. So endoneurium will cover an axon. Perineurium covers a fascicle, and a fascicle is a collection of axons. So if you've done your skeletal muscle, it's very similar. So when I look on this entire nerve to the right here, all of this is the nerve, and I break it down into segments called fascicles. So this is one fascicle, this is another, and then another here. So a nerve is a bunch of fascicles. Remember I said there's a bunch of axons, but we can go deeper into it. So when I look inside a fascicle, all of these tiny little circles inside are axons. And sometimes they'll give that wider appearance because of the myelin. But what I can't see, if I went on a higher magnification, all these tiny axons are going to be covered with Endoneurium. So I cover with Endoneurium, and then I take a bunch of axons and I wrap it with Perineurium, and the perineurium is what forms a fascicle. And then I take a bunch of fascicles and I wrap them together with Epineurium. So all of this is connected together, and this collection of these different layers is all connective tissue. So I'm using endoneurium to support an axon, I'm using perineurium to support a fascicle, and then epi towards the edge. Epi supporting the entire nerve. So it's straws within a straw. just like we did skeletal muscle. So when we look at a nerve, you're focusing on the layers that create the different pieces to it. So a fascicle is a bunch of axons. Put a bunch of axons together. I form that fascicle and wrap you with perineurium. I take a bunch of fascicles and I create the entire nerve and wrap it with epineurium. This is nerve histology. And then we looked at neuron histology.